Recently, the journal Nature published a study titled Whole Genome Ancestry of an Old Kingdom Egyptian by Morez Jacobs and colleagues. This study claims to provide groundbreaking insights into ancient Egyptian ancestry. This research represents not scientific advancement, but rather a profound failure of basic scientific standards and ethical research practices. What I present today is not merely criticism, but a systematic exposure of a study that fails to meet even the basic requirements of a valid scientific research. The implications extend far beyond academic debate, affecting how we understand African contributions to world civilization. Let us begin by examining what the authors claim in their introduction. Here, we present the first complete genome of an ancient Egyptian from the Old Kingdom period, providing unprecedented insights into the population history of ancient Egypt. Our analysis reveals that this individual possessed approximately 80% of North African ancestry and 20% West Asian ancestry, suggesting significant population movement from the Fertile Crescent into Egypt during the formative period of Egyptian civilization. This bold claim exemplifies the fundamental problem with this study, drawing sweeping conclusions about an entire civilization based on woefully inadequate evidence. Let me explain why this approach fails basic scientific standards. The most glaring failure of the Nuarat study is its attempt to characterize the genetic makeup of an entire civilization spanning over 4,000 years based on a single individual. This violates the most basic principles of scientific research. Ancient Egyptian civilization encompassed millions of people across a vast territory, from the Mediterranean to Nubia, with significant regional and temporal variations. The notion that one person's DNA could tell us about the entire civilization is as absurd as claiming that analyzing the DNA of one modern American could tell us about the genetic makeup of all Americans throughout US history. The authors themselves acknowledge this limitation, but then ignore its implications. Quote, "While we acknowledge that a single genome cannot capture the full genetic diversity of ancient Egypt, our results nonetheless provide valuable insights into the population history of the region during this critical period of state formation." Unquote. This statement reveals extraordinary logical inconsistency. The authors correctly admit that one genome cannot represent population diversity, yet then claim this same inadequate sample, however, provides valuable insights into population history. In any legitimate scientific field, attempting to characterize an entire population based on a sample size of one would be immediately dismissed. Yet, the journal Nature published this study, raising serious questions about their scientific standards. Even if we momentarily set aside the problem of using one individual, the specific person they chose introduces multiple layers of bias that further undermine the study's conclusions. For example, the authors describe the burial site as follows. Quote, "The individual was recovered from a rock-cut tomb at Nuarat, approximately 265 kilometers south of Cairo. The burial contained an elaborate ceramic vessel in which remains were interred, along with copper tools and important ceramic vessels suggesting high social status and possible foreign connections." Unquote. This description itself raises immediate red flags about how representative this sample could be. The location in Middle Egypt was a known corridor to trade and foreign contact. The elaborate burial indicates elite status and potential foreign connections. This is precisely the type of individual who would be least representative of the general Egyptian population. What the authors conveniently fail to acknowledge is the overwhelming archaeological and historical evidence pointing to the southern origins of ancient Egyptian civilization. The cultural heartland of ancient Egypt was not in the north, near the Mediterranean, or the east, near Asia, but in the south, in Upper Egypt and Nubia. The earliest evidence of Egyptian state formation comes from sites like Hierakonpolis, Nagada, and Abydos in Upper Egypt, much further south than Nuerat. Had the researchers been genuinely interested in understanding the genetic makeup of ancient Egyptians during the formative period of Egyptian civilization, they would have prioritized sampling from Upper Egypt and the Nubian region. The pharaohs of the Old Kingdom, particularly those who ruled from Thebes, modern-day Luxor, would have provided a much more relevant genetic profile for understanding the origins of Egyptian civilization.
Ancient Nubia and ancient Egypt share roots, common cultural roots in Northeast Africa, in the Sahara, and along the Nile Valley. The ancient Egyptians and the ancient Nubians can both be described, especially in the earlier periods, as being descendants of Northeast African indigenous populations. As one writer said, all of these people are Africans, and I define Africans as people whose biological histories have emerged and their identities have emerged in Africa, and where any mixing that took place on African soil. The Egyptians did not come from any place else other than the Nile Valley. Their identities and any other ancestry that they may have had were all forged together in that space.
These rulers represented the continuation of indigenous African traditions that gave birth to Egyptian civilization, not foreign-influenced elites from trade corridors. This sampling bias is particularly egregious given the well-documented southern origins of Egyptian kinship, religious practices, and cultural symbols. The white crown of Upper Egypt predates the red crown of Lower Egypt, and the unification of Egypt was achieved by southern kings expanding northward. The most sacred sites and earliest monumental architecture are concentrated in the south. By choosing a sample from Middle Egypt with clear elite and potentially foreign connections, the authors have deliberately selected an outlier rather than a representative sample. This would be like trying to understand the genetic makeup of modern Americans by sampling only from a wealthy international businessman buried in New York City with imported luxury goods. Such a sample will tell us very little about the broader American population. The Nuerat study is not the first to employ such biased sampling to distort ancient Egyptian history. In 2016, Johannes Krauss and colleagues published a similar study that claimed to provide insights into ancient Egyptian genetics. The study primarily used samples from Abu Sir Melek, a site known for Greek and Roman influence during the period sampled. Most of their samples dated to the Ptolemaic and Roman periods, around 332 BCE to 426 CE, when Egypt was under foreign rule. Yet they made sweeping claims about ancient Egyptians in general. The Krauss study committed the same fundamental error as the Nuerat study, using unrepresentative samples to draw sweeping conclusions about an entire civilization. They sampled individuals from a northern site during periods of foreign occupation, then expressed surprise at finding genetic similarities to Eastern Mediterranean populations. Both studies systematically avoid sampling from the regions and periods that would have most likely revealed the indigenous African origins of Egyptian civilization. The avoidance of Upper Egyptian and Nubian samples is not coincidental at all, but represents a deliberate sampling strategy designed to minimize evidence of African origins. Yet the authors make this exact unwarranted leap. Quote, "The genetic profile of the Nuerat individual provides a window into the ancestral composition of ancient Egyptians during the formative period of state development," suggesting that West Asian gene flow played a significant role in the population history of ancient Egypt, unquote. This statement fundamentally misrepresents what can be concluded from analyzing a single elite individual. The genetic profile of one wealthy person tells us nothing about the ancestral composition of ancient Egyptians as a whole. This is not merely a minor limitation, but a fatal flaw that invalidates the entire study's conclusions. The study's approach to analyzing ancestry represents a masterclass in data manipulation. The authors have constructed a framework that systematically biases results toward non-African ancestry through several questionable methods. Consider how they describe their reference populations. Quote, "For our ancestry analysis, we used a curated set of ancient and modern reference populations from North Africa, West Asia, and the Eastern Mediterranean. These included Neolithic and Chalcolithic populations from Anatolia, the Levant, and Mesopotamia, as well as modern North African populations including Berbers, modern-day Egyptians, and Nubians. This seemingly neutral description hides critical problems. First, the study artificially separates North Africa from African populations, creating a false division that has no basis in historical reality. This categorization imposes modern political boundaries onto ancient genetic data. The authors then state, "Our admixture analysis indicates that the Nuerat individual's genome can be modeled as a mixture of approximately 80% North African related ancestry and 20% West Asian related ancestry, with the latter component showing closest affinity to ancient populations from the Eastern Fertile Crescent." This statement reveals circular reasoning. The categories of North African and West Asian are not objective genetic classifications, but imposed geographic constructs. By establishing these categories beforehand and then assigning ancestry percentages to them, the authors create an illusion of scientific precision while actually engaging in a form of statistical gerrymandering. Second, the study heavily weighs its reference populations toward non-African sources, while inadequately representing the diverse African populations that would have constituted the primary source of indigenous ancient Egyptian ancestry. The systematic exclusion of appropriate reference populations from Nubia, the Eastern Sahara, and other regions of Africa with documented historical connections to ancient Egypt represents not an oversight, but a deliberate manipulation of the comparative framework. Third, the study compares the Nuerat genome to reference populations separated by thousands of years in time. Quote, "For ancient West Asian reference populations, we included individuals from Neolithic Anatolia, dating 8000 to 6000 BCE, Chalcolithic Levant, 5000 to 3500 BCE, and Bronze Age Mesopotamia, 3000 to 1000 BCE. This temporal mismatch renders genetic comparisons meaningless. It will be like comparing your DNA to people who lived thousands of years before or after you to determine your ancestry. Such comparisons tell us nothing about recent population movements or influences. These manipulations transform what should be a neutral scientific analysis into a politically motivated narrative designed to minimize African contributions to Egyptian civilization. The statistical analysis presented in the Nuerat study uses impressive-sounding techniques to create an illusion of scientific rigor around fundamentally flawed data. For example, let's examine some specific claims. Quote, "Principal component analysis, or PCA, of the Nuerat genome alongside reference populations shows clustering with modern North African populations, but with a slight shift toward ancient West Asian populations." Unquote. This analysis contains multiple problems. that would be obvious to any trained statistician. First, using principal component analysis with a single sample point makes no scientific sense. PCA is designed to identify patterns across multiple samples. Applying it to a single sample produces meaningless results. The coordinates provided create an illusion of precision while actually representing a statistically invalid approach. The presentation of ancestry percentages to the first decimal place, complete with error estimates, creates an impression of scientific rigor that is entirely unwarranted. The error estimates themselves are meaningless, as they only measure technical variability in the analysis rather than the fundamental uncertainty introduced by using a single sample and a biased reference population. Moreover, the author's decision to use 4 ancestral components represents an arbitrary choice that fundamentally shapes the results. Different values would produce entirely different ancestry percentages. This arbitrary selection represents not legitimate scientific methodology, but rather a form of cherry picking, designed to produce results that align with the author's predetermined narrative. The author's statistical analysis is not merely flawed, but fundamentally misleading. It employs sophisticated statistical methods inappropriately and uses precise numerical values to create an illusion of scientific scrutiny or rigor around conclusions that the data simply cannot support. Perhaps the most troubling aspect of the NeuroRat study is how it contradicts well-established scientific understanding of human origins and migration patterns. The study's framing and conclusions fundamentally misrepresent the direction of human migration in Africa and the Middle East. The authors state, quote, "Our findings suggest significant gene flow from the Fertile Crescent into Egypt during the formative period of Egyptian civilization, potentially associated with the introduction of advanced agricultural techniques, administrative practices, and religious concepts that contributed to the development of the Egyptian state," unquote. This statement directly contradicts the overwhelming scientific consensus on human migration patterns. The genetic, archaeological, and paleontological evidence clearly establishes that the primary direction of human migration was out of Africa, not into it. Humans evolved in Africa approximately 300,000 years ago and subsequently spread to other continents. This fundamental fact means that genetic similarities between African and non-African populations primarily reflect African ancestry in non-African populations, not the reverse. The authors' own data actually support this established pattern, though they deliberately misinterpret it. Quote, "Analysis of mitochondrial DNA haplogroups reveals that the NeuroRat individual belonged to haplogroup L3, which originated in East Africa approximately 60,000 to 70,000 years ago, and gave rise to the non-African haplogroups M and N," unquote. This finding directly contradicts the author's narrative of significant West Asian influence on ancient Egypt. Haplogroup L3 is an African lineage that represents one of the ancestral populations from which all non-African populations are derived. Its presence in the NeuroRat individual provides strong evidence for indigenous African ancestry. Similarly, the authors report, quote, "Y chromosome analysis indicates membership in haplogroup E1b1a, which has its highest frequency and diversity in so-called sub-Saharan Africa," unquote. This finding further contradicts the author's narrative. Haplogroup E1b1a is predominantly found in sub-Saharan African populations and represents an indigenous African lineage. Its presence provides additional evidence of African ancestry rather than West Asian admixture. Quote, "While the uniparental markers show African affinities, the autosomal analysis reveals a more complex ancestry profile with significant West Asian components," unquote. This statement represents a deliberate attempt to minimize evidence that contradicts their preferred narrative. The phrase, quote, more complex ancestry profile, unquote, serves as a rhetorical device to privilege their questionable autosomal analysis over the more straightforward and reliable evidence from mitochondrial DNA and Y chromosome analysis. The study's fundamental misrepresentation of human migration patterns represents not legitimate scientific interpretation, but rather the perpetuation of colonial narratives that systematically minimize African contributions to world civilization. The study's approach to verifying the authenticity of ancient DNA falls far below basic scientific standards. The authors provide vague descriptions rather than specific details about their methods. For example, quote, DNA was extracted from a Petrus bone using established protocols for ancient DNA recovery. Standard precautions against contamination were employed, including physical separation of pre- and post-PCR environments and the use of negative controls, unquote. Well, this description lacks the specific details required for proper evaluation in any scientific paper. The phrase standard precautions obscures the actual procedures used. In legitimate ancient DNA research, detailed documentation of contamination controls is essential in any paper because ancient samples are extremely vulnerable to contamination from modern DNA. The authors further state, authentication of the ancient DNA was confirmed through observation of characteristic damage patterns consistent with ancient DNA, including C to T transformations at the 5' ends of DNA fragments. Well, this represents a minimal and insufficient approach to authentication. While damage patterns are one marker of ancient DNA, proper authentication requires multiple independent lines of evidence. The study's authentication protocols appear inadequate by current scientific standards. The authors provide similarly vague information about the analytical methods. For example, quote, sequencing data were processed through standard bioinformatic pipelines for ancient DNA, including adapter trimming, mapping to the human reference genome HG19, and filtering for PCR duplicates and mapping quality, unquote. Well, this description lacks the specific parameters and analytical decisions necessary for other researchers to verify the results. The phrase standard bioinformatic pipelines obscures the actual procedures used, making it impossible for other scientists to replicate or verify the findings. This lack of transparency violates a fundamental principle of science, which is what? Reproducibility. If we cannot reproduce your study, it is a lie. These failures in basic laboratory and analytical methodology would be sufficient to disqualify the study from publication in any legitimate scientific journal. Their presence in the study published in Nature raises serious questions about the journal's review process and editorial standards. The study's most intellectually dishonest aspect is the systematic suppression of the overwhelming archaeological, anthropological, linguistic, and artistic evidence that contradicts its conclusions. The authors state, quote, the presence of a significant West Asian ancestry component in the Nuerat's genome suggests that the development of Egyptian civilization was influenced by population movements from the Fertile Crescent, potentially bringing agricultural technologies, administrative practices, and cultural innovations that contributed to the formation of the Egyptian state, unquote. This statement conflates genetic ancestry with cultural diffusion, a fundamental error in archaeological interpretation. The presence of genetic similarities, themselves questionable given the methodological flaws, provides no evidence whatsoever for the transfer of agricultural technologies, administrative practices, and cultural innovations. This leap from genetic data to cultural diffusion represents not legitimate specific scientific inference, but rather the imposition of a predetermined narrative. The authors further state, our findings align with archaeological evidence for connections between Egypt and Mesopotamia during the late pre-dynastic and early dynastic periods, including similarities in artistic motifs, architectural elements, and administrative technologies such as cylinder seals and writing systems. This statement misrepresents the archaeological evidence, which actually demonstrates indigenous development of these cultural elements within Africa. The archaeological record clearly shows that writing systems, monumental architecture, and complex administrative practices developed independently in Egypt through continuous evolution from earlier African traditions. Consider the author's brief acknowledgement of physical evidence. Physical anthropological analysis of the Nuerat individual suggests that he likely had brown eyes, brown hair, and skin pigmentation ranging from dark to black skin. This description is entirely consistent with indigenous African ancestry, yet the authors fail to address how this physical description aligns with their claim of significant West Asian ancestry. This selective presentation of evidence represents profound intellectual dishonesty. The study makes no mention of the linguistic evidence that places ancient Egyptian within the African branches of the so-called Afro-Asiatic language family, with its closest relationships to African languages. This linguistic evidence directly contradicts the study's implications of significant West Asian influence. Similarly, the study completely ignores the thousands of artistic representations in which ancient Egyptians depicted themselves with distinctively African physical features. These representations provide direct testimony to ancient Egyptian self-identification and directly contradict the study's claims about Egyptian ancestry. The archaeological record provides overwhelming evidence for the indigenous African development of Egyptian civilization, with continuous cultural evolution from early hunter-gatherer societies through to the unified Egyptian state. This development shows clear continuity from earlier African cultural traditions. The study's failure to engage with this contradictory evidence reveals its fundamentally unscientific nature. Scientific inquiry requires researchers to consider all available evidence and to address contradictions in their interpretations. The systematic suppression of contradictory evidence represents not legitimate scientific practice, but rather the imposition of predetermined conclusions regardless of evidence. The publication of the Nuerat study raises serious questions about the integrity of the scientific review process. Any competent reviewer with basic knowledge of population genetics or Egyptian history would have identified the study's fatal flaws and rejected it immediately. The author's acknowledgements section provides disturbing insights into the institutional context of this study. This research was supported by grants from the European Research Council, the Max Planck Society, and the British Museum's Ancient DNA Research Initiative. These funding sources raise serious questions about institutional biases. European research institutions have long histories of supporting research that minimizes African contributions to world civilization. The British Museum, in particular, has a vested interest in narratives that separate ancient Egyptian civilization from its African context. As such, narratives help justify its continued possession of looted Egyptian artifacts. The fact that the study was nevertheless published suggests either reviewer incompetence, editorial bias, or outside interference in the review process. None of these explanations is compatible with legitimate scientific publishing. The author's media engagement following publication reveals the political dimensions of this research. In interviews with major media outlets, lead author Dr. Maurice Jacobs emphasized that the study provides scientific evidence for the complex interactions between Africa and the so-called Near East that shaped ancient Egyptian civilization, adding that while Egypt is geographically in Africa, its cultural and genetic connections to West Asia were also significant. This framing exemplifies the subtle rhetorical strategies used to minimize African contributions to Egyptian civilization. The phrase, while Egypt is geographically in Africa, implicitly separates Egypt from its African context, suggesting that its physical location on the African continent is somehow incidental rather than fundamental to its identity. This rhetorical strategy represents not legitimate scientific communication, but rather the perpetuation of colonial narratives through carefully crafted language. The Nuerat study represents not an isolated incident of poor methodology, but rather the continuation of a long tradition of using pseudo-scientific methods to support colonial narratives about African history. This tradition includes 19th century skull measurements and scientific racism, mid-20th century blood group studies, and late 20th century attempts to use limited genetic markers to support non-African origins for ancient Egyptians. The persistent attempts to deny the African origins of Egyptian civilization cannot be understood without recognizing the economic stakes involved. And what do these include? The multi-billion-dollar Egyptian tourism industry, largely controlled by European and American companies. The legitimacy of European and American museums' continued possession of looted Egyptian artifacts. The substantial funding available for research that supports Eurocentric historical narratives. The profitable media ecosystem that continues to present ancient Egypt through a Eurocentric lens. This weaponization of genetics represents not legitimate scientific inquiry, but rather the continuation of colonial knowledge production by technological means. Any legitimate genetic study of ancient Egyptian populations would need to meet standards of scientific validity. And what do these include? Adequate sample size with dozens or hundreds of individuals from diverse regions, time periods, and social contexts. Appropriate reference populations that are temporally and geographically relevant. Transparent methodology with complete documentation of laboratory procedures. Integration with archaeological, anthropological, linguistic, and artistic evidence. Appropriate statistical methods with explicit consideration of limitations. A neutral interpretive framework that does not privilege particular historical narratives. Independent verification by researchers not involved in the original study. The Nuerat study fails to meet any of these basic standards, rendering it scientifically worthless, regardless of its publication in a prestigious journal like the journal Nature. Moving beyond the pseudo-scientific approach requires a fundamental transformation of genetic research on ancient populations. This includes research led by African scientists and institutions, meaningful involvement of descendant communities, true integration of genetic research with other disciplines, full transparency regarding funding sources, complete sharing of raw data and methods, and explicit acknowledgement of the historical context of genetic research. Until these conditions are met, studies like the Nuerat's Genome Project will continue to represent not scientific advancement, but rather the perpetuation of colonial mythology through pseudo-scientific methods. The Nuerat Man Genome Study represents a complete failure of scientific methodology, interpretive integrity, and academic ethics. Its conclusions are not merely questionable, but entirely invalid from a scientific perspective. The study should be retracted immediately, and its publication should prompt a thorough investigation of Nature's review and editorial processes. The ancient Egyptians built the pyramids, developed hieroglyphic writing, made fundamental advances in mathematics and astronomy, and created one of the world's most enduring civilizations. They did so as African peoples, drawing on thousands of years of indigenous African cultural traditions and innovations. This historical reality is supported by overwhelming archaeological, anthropological, linguistic, and artistic evidence that cannot be erased by methodologically bankrupt genetic studies. The Nuarat study represents not a scientific contribution, but rather a profound scientific failure, a case study in how not to conduct genetic research on ancient populations. Its publication damages not only our understanding of ancient Egyptian history, but also the credibility of genetic research more broadly. Only by recognizing and rejecting such pseudoscientific approaches can we begin to develop a truly scientific understanding of ancient population histories that represents both methodological rigor and historical agency of African peoples. As scholars committed to intellectual integrity and historical truth, we must demand better from our scientific journals, our research institutions, and our colleagues. The struggle for historical truth about African civilization is far from over. We must remain vigilant against attempts to use pseudoscientific methods to deny the African origins of one of humanity's greatest civilizations. Thank you for your attention to this critical matter of scientific and historical integrity. Remain black and proud.
We begin our journey in the Great Lakes region of Africa. The Anu, migrants from the Great Lakes region, were the first settlers of the Nile Valley. The first written scripts were found in Abydos and dated to about 3,400 BCE. These predate the cuneiform script of Sumer by at least 2 centuries. The Scorpion King was a ruler of Upper Kemet in the pre-dynastic period. The first dynasty covers the first series of kings to rule over a unified Kemet, the Old Kingdom. It is also known as the Age of the Pyramids, encompasses the reigns of the great pyramid builders such as Khufu and Khafra. The Kerma culture was based in the southern part of Nubia, or Upper Nubia, in parts of present-day northern and central Sudan. The Middle Kingdom started from the reunification of Egypt under the reign of Mentuhotep II in the 11th dynasty to the end of the 12th dynasty. Thutmose III attacked Nubia, but only went so far as the 4th cataract of the Nile. The new kingdom included some of Kemet's most famous kings, including Ahmose I, Hatshepsut, Thutmose III, Amenhotep III, Akhenaten, and Tutankhamun. 25th dynasty, or the Napatans, after their capital Napata. Notable pharaohs include Shabaka, Taharka, Shebitku, and Tantamani. Kemet had fallen by this time due to various invasions: Assyrians, Persians, Greeks. Through the conquest of Alexander the so-called Great, Romans, and in more recent times, the Arabs whose descendants currently occupy a land that has been renamed Egypt by colonizers. Until the lions have their own historians, the tales of the hunt will always glorify the hunter.